All right, it's exactly noon here in Ann Arbor, Michigan. Good day, everybody. And it is my pleasure to introduce Dr. Mary Scapo. But before I start with the introduction, I would like to remind everybody that there will be two talks today. The second one will be given by Dr. Stefan Rüdiger. And each talk will be about 30 minutes, followed by about 10 minutes for questions and answers. We can now move to the introduction for the sake of time to Dr. Stefan Rüdiger. He's currently head of the Department of Chemistry, and Dr. Rüdiger graduated in chemistry at Heidelberg University, and he obtained a PhD from Freiburg University, summa cum laude. After a postdoc with Dr. Alan First, Cambridge University and Medical Research Council, and supported by a long-term fellowships of EMBO and Marie Curie program, he started his own group at Utrecht University. Dr. Rüdiger is Marie Curie Excellence Team Leader and high potential at Utrecht University. His work centers on the molecular mechanisms of protein folding and chaperoning in the cell, and with a particular focus in understanding the mechanism of chaperone machines. Thank you so much for coming today, and I don't want to take much of your time.
Thank you so much for your kind introduction, and I try to share my screen. So I show some data on the chaperoning of aggregation diseases, and which means I will first start with explaining mechanisms on chaperones, and then we will look at aggregates. Now, one way look at aggregates, we by now that all the various aggregates you find in diseases, they do have completely different shape, redefined shape. We know how they look like. We can now look at an example at Alzheimer's disease. When we patients in Alzheimer, they do get a shrinkage of the brain. And the origin of that brain shrink is protein aggregation. You have actually an aggregation event outside the cell from the A-beta peptide. And this triggers somehow via unknown mechanism aggregation of tau inside the cell. So now chaperones do act inside the cell. And if you want to kill a cell, basically also you do have act inside the cell. And my aim is now actually to look at these processes that lead to the aggregation, basically from the monomer to the fibril. And that what might be the most interesting species is potentially that what's in the middle. Yeah? The early intermediates that actually trigger the formation of the fibril. Now, we now think this is a process that takes place in disease. And we now do have all in our cells molecular chaperones. And these molecular chaperones, they are in principle quite efficient, right? When you now think this process of protein aggregation, this takes place could potentially take place in all of our cells. We all have a lot of these proteins that do aggregate, such as tau or alpha-synuclein. And so now you wonder, okay, maybe these proteins would not aggregate in our brain, but in fact, you can actually trigger this aggregation, and with, depending on conditions, within a day or a week, it would aggregate. So the fact that after this seminar, you may still be able to remember the presentation, this might be related to the fact that we do have these molecular chaperones, which under normal conditions really prevent this aggregation from happening. So this means before we now look at the aggregation process, we have to understand how actually do chaperones act? And so to basically answer this question, if you have so many chaperones, why do we get fibrils? So basically, though, I would like to start. How do chaperones fold proteins? Basically, this means starting with an unfolded protein and see how chaperones shift this protein towards unfolded protein. So when we look at the folding pathway, it's actually quite simple sequence. We at the end of the folding pathway, we end up with a native state that's active, enzymatic activity. The potential can measure that. To get there, proteins do fold via folding intermediates. And in these folding intermediates, the first step is actually proteins bury the most hydrophobic side chains to form this hydrophobic core. Then totally unfolded protein, these yellow hydrophobic stretches, they would all be exposed. And around these hydrophobic stretches, the protein folds. And around this more hydrophobic core, we have an outer shell of hydrophobic residues, which are stretches that are not as hydrophobic compared to that what does fold that core. And they later oriented around the folding nucleus. So now we do have inside the cell conserved families of molecular chaperones. So these families are conserved from bacteria to man, and typically we have several parallels of each of these chaperone families, but the most important ones are the HSP70 and HSP90 family. Quantitative most important, but also functionally they're most important. And I would now like to start looking into chaperone mechanism by looking at the HSP70 family. The HSP70 family is an ATP-triggered machine. So in an ATP-bound form, at the right top, the substrate binding site is open. An unfolded protein could simply bind to the substrate binding site of HSP70. So, but as the binding site is open, the protein can dissociate. So the trick of the machine is that you can hydrolyze the ATP to ADP, and if that happens, you basically enclose the substrate in the substrate binding site. And to do that, okay, we do have an ATP cycle. Having an ATP cycle has the advantage that you can trigger the action of the chaperone machine, so that hydrolysis of ATP is triggered by J-domain proteins and by nucleotide exchange factors. So the nucleotide exchange factor exchanges the ADP in the substrate bound form back to ATP, and the substrate can be released. So that's the basic mechanism of this machinery. Now we can measure the enzymatic activity. We can measure, for example, the enzymatic activity of HSP70 by taking a folded protein. Here on the right, you see firefly luciferase. Firefly luciferase is a protein that emits light when fully folded. You can easily unfold luciferase either by thermal denaturation or by chaotropic reagents such as Rüdiger or guanidinium chloride. So now, we can measure the luminescence of the luciferase, and we can, for example, increase now the concentration of an HSP70 machinery and look, can it refold the protein? Now, titrate in here HSP70. Actually, we use the E. coli system, which is called DNAK. And we use, I explained to you in the picture before, right? There are co-chaperones, a J-domain protein, hydrolyzing ATP, and a nucleotide exchange factor, which is now in cola here called GAPE. They form a machinery, and as you can see, they can refold luciferase. If you put in HSP70, you get more refolded luciferase. It's a reaction that takes about 3/4 of an hour. So, when we look at that, how can we understand that? Basically, we have an unfolded protein that you get when you actually unfold the protein guanidinium chloride. The protein has two choices. Yeah? So basically, the protein can take the pathway to the native state, or, and that's actually the majority for this luciferase protein, it would enter a pathway in which you end up with oligomers or which states which do not immediately fold on their own. So what happened then is that you have some molecular chaperone HSP70 that grabs this folding intermediate and puts it back into another folding cycle. That is the result, what you see here, right? You can refold the luciferase quite efficiently. However, if you now increase the HSP70 concentration further, you should say, yeah, I mean, if I have more and more, it should be better and better. But in fact, if we increase the HSP70 concentration further up to 10 micromolar, what we do see, the activity of this enzymatic activity of luciferase is basically back to the beginning. So, and what happens here, that you may have several HSP70s binding to the folding intermediates that's never released again, and it doesn't have a chance to fold. So now this seems to be, on the one hand, quite unfortunate. On the other hand, you may argue, okay, if it works effectively at 3 micromolar, why pipetting in 10? Yeah, fair enough. However, when we look how what's actually happening here, we have to first understand why doesn't it go further. Yeah. So now when we look at is a HSP70 substrate binding pocket. Yellow are the side chains that are hydrophobic, and what you see when you zoom in, it's all hydrophobic. I've shown you before how it looks when a folding intermediate exposes itself to the outside. Yeah, you have a lot of hydrophobic residues that form the hydrophobic core, and that's recognized by HSP70. So HSP70 bind to the very exact residues that do form this hydrophobic core. So now, if you put in a lot of HSP70, such as what we do have here, 10 micromolar, then actually the hydrophobic core around which the protein folds cannot form anymore. So, and that's why you get this folding. So now, good. The interesting aspect is that you say, okay, why pipetting at 10 micromolar if 3 micromolar work? In fact, the physiological concentration of HSP70 can be up to around 30 micromolar, and at heat shock even more. So we are basically very much to the right outside of the screen here. So now then we seem to have basically an efficient system, but there might be a solution. And that solution is that there is also the molecular chaperone HSP90, which is known to act often downstream of HSP70. It's also a machine that actually runs with ATP. So if you now work at 10 micromolar, then, and you add at that very concentration, you also add HSP90. That's what we have here on the right. So then we see the moment we add HSP90, we immediately go up with activity, despite having 10 micromolar HSP70, right? So basically what happens is that the HSP90 takes over the substrate from HSP70 and allows the protein to fold on HSP90 to reach the native state. So basically what HSP90 does functionally, it dissolves the folding block of HSP70. And while I have shown that now, actually with the E. coli system, you can do the same experiment with the human system, and that is basically what you see here. You see the same folding block if you titrate in HSP70, it blocks the folding, and if you now titrate in HSP90, what you do see, you go up this activity. So this is a conserved process for these two chaperone systems. So basically what we can learn from that is that we have actually 3 productive folding pathways. There is the yellow circle, it's basically spontaneous refolding. You do have the green circle, there's HSP70 cycling. So basically the HSP70 binds a folding intermediate, and then it has the option either after release it folds to the native state, or it doesn't work. Then you basically release a protein that can fold again, and you basically can cycle. Or you have the highway, and the highway works with HSP90. So basically the protein can go from the HSP70 to HSP90, and from there directly go to the native state, basically without the cycling. And by that, you actually cut the dead end pathway where a lot of HSP70 would bind to the substrate and prevent the folding of the protein. So basically, it works like that. The HSP70, as I indicated before, is a very hydrophobic a substrate binding pocket triggered by ATP, and it recognizes the most hydrophobic parts of the protein at first, and then it transfers to HSP90, which allows the formation of the hydrophobic core. Now, these processes are very early, and I haven't shown you now the data, but if you would inhibit HSP90 to block that, you would see that if you add the inhibitor only, let's say after 2 minutes of the reaction, or even immediately with HSP90, it doesn't inhibit that reaction anymore. And you pass a point of no return, the point of no return, when the protein already starts folding on its own. And that is actually the longest part of the folding trajectory. The protein folds on its own after getting out of the claws of the chaperone machine. So basically, the proteins do fold in the early stages. If you illustrate it a bit more artistically, you have the red hot phase of protein folding when the chaperones act. When they have acted, then the protein can travel on the energy hypersurface very slowly to its native state. And it does so on its own. It doesn't do so with then the further assistance of the chaperone machinery. Good. This is how the chaperone machinery works. And we do learn from that HSP70 and HSP90, they together form a folding cascade, a very efficient folding cascade. The question now is, can we use our understanding on the chaperones now? We look at protein aggregates, which are introduced at the beginning, which would basically mean we start with the aggregate and we try to go to the green corner at the top right of the picture. Good. Now let's have a look how aggregates look like. I mean, the laboratory of Schor's Scheres provided Cryo EM structures for lots of various fibrils. And we now look at here one example. It's actually the fibril from Alzheimer, fibrils, and from material purified from patient's brain. So what we do see, we basically have a dimeric structure, right? So that's one half, and you see here is the other half, and it's basically a dimer of two fibril strands, right? So here you see on the right a fibril strand, and you see here one on the left. So and then we have all the layers of the fibrils. Now, this is the core of the fibril. In the previous talk, you have heard about intrinsically disordered protein. In fact, tau is an intrinsically disordered protein. It only adopts a shape when it does form these fibrils, but the largest part of tau is still hanging outside. So basically at the N terminus and at the C terminus, we have long stretches of disordered parts of the protein. So now, when we look at that part of the tau protein, it's actually quite interesting to see that we do have two HSP70 binding sites, one at the end of the fibril, one in the disordered part of the protein. So now we wonder, can we actually disaggregate these fibrils with HSP70 machinery? And so what we did is we took, you see here, actually indicated the entire sequence of tau, but in fact, the part of the protein which is aggregation prone, it's a microtubule binding domain. This is boxed here, and this microtubule binding domain, you also can purify that fragments. It gets faster. This is a full-length protein. You see on the right, you see negative staining EM pictures of such fibrils. Now we wonder if we make these fibrils, which you see on the right, can we disaggregate them with chaperones? And to visualize that, we use density gradient, sucrose gradient. So basically you have a gradient from 10 to 40% sucrose. And when you now separate your mix of monomers, oligomers, fibrils, you separate it, the fibrils go to the bottom, the monomers swim on top. So, and this you can visualize, for example, with dot plots. Then you see the monomer on top, and you see the fibrils at the bottom. Now with this technique, it's you now can see, good, we take fibrils, we add the HSP70 chaperone machinery, and look what happens. So when we do that, we do see in these dot plots, when we add HSP70 and ATP together, yeah, that's the third lane from the left, we do see on top that we get protein back on top, while the fibrils stay at the bottom. If we leave out ATP, then we don't see any disaggregation. So very good. You should think, yeah, that a human system can disaggregate these tau fibrils. You can quantify these plots, and then indeed you see, yeah, only with ATP and the HSP70 system you can disaggregate these fibrils. Good, great. Should be happy with that. But there's a but. Now that but is that becomes evident, this but, when we look at the structure of various fibrils. And said before, right, so the laboratory of Shaw Scherers, they solve patient-derived structures of lots of fibrils of various tauopathies. So now we have at the top, you see fibrils derived by Alzheimer. You have two different types of fibrils, parahelical filaments, which I showed before, but also straight filaments. They basically have the same fold, just the dimerization of the fibrils is different. For fibrils of Pick disease, we have fibrils of corticobasal degeneration. Fine. And at the very bottom, you see artificial heparin-induced fibrils. So heparin-induced fibrils, you add heparin. That's how we made these fibrils I showed you before of the microtubule binding domain fragment. You can induce in vitro fibrils. These fibrils, they look very different than the fibrils you obtain from patient material. They're much, much smaller. And in green, we do see the HSP70 binding sites, right? I indicated them to you before in the Alzheimer fibril. And these stretches that bind HSP70, when you look at them where they are, they give you some orientation about the difference of these fibrils, no? and the different shapes. Good. Question is, if something works for artificial fibril, which you see at the bottom, would it actually work for real fibrils? Would it work for patient fibrils? So that's our question. Can HSP70 chaperones disaggregate patient fibrils? Now, the question is also quite justified when we look where in the patient, when we look at the patient material, what part of tau is in the patient fibril. Then we do see that the actual Alzheimer fibril is only a part of the microtubule binding domain. So the artificial fibrils we make, they comprise the entire microtubule binding domain. But there's a stretch at the very end of the Alzheimer fibrils that's lacking in the microtubule binding domain. And the Alzheimer fibril lacks the first two repeat regions. Now, what does it mean? Now, basically, if the end of the fibril is lacking, you lack the part that is actually pairing the HSP70 binding site. So such a fibril would not be stable. So this means the patient fibril definitely needs and must look different than the artificial fibril. We've seen that before, right? They are different, but we can understand that it must be also a reason that there is a completely different build. So what does this mean? Now, what this means is that we actually now purified fibrils from various from various tauopathies. And we now looked at what do we see if you look for this aggregation if we do that. And what we see is, and this is now we looked at this on gels, then we can see on the left the gels plus minus ATPase, various Alzheimer, Pick's disease, and cortical basal degeneration with HB70 cocktail and plus or minus ATP. And now looking at the difference, do we see a difference? Well, the gel looks slightly messy. If you quantify it, what do you see? Well, not much of a difference. So now this is not really a high resolution method. The question is, can we get more resolution to it? And for that, I would like to explain you a method we are now using to investigate that. And so what we are doing is we are now looking at the process from monomer to fibril. And we are doing this now with two different aggregates. We do this with a tau monomer. We talked about this all the time. We're also now doing this actually for the Huntington fibrils. And so you have different ways how you make the fibrils. Yeah, in Huntington, you produce the protein as a fusion. You cleave the fusion protein, then you get the Huntington protein, and you can monitor, for example, and it's a standard assay in the field, you can measure the increase of fluorescence over time and the formation of the aggregate with a fluorescent dye, which is called THT, thioflavin T. So now, and with tau, we induce the aggregation with chaperone. And then again, we monitor the same thing, THT fluorescence. Basically, it's a thing for basically all sorts of aggregates, right? So you get an increase in fluorescence. When the protein aggregates. Good. But now how can we monitor that and quantify that at high resolution? And for that, we now used a relatively novel technology, flow-induced dispersion analysis, called FIDA. And the basis of this is that you have a mixture, an unlabeled molecule, so-called analyte, on labeled molecule, an indicator. And this mixture of an indicator and an analyte, you shoot through a capillary of 75 micrometer. And now in a capillary, when you shoot something through a capillary, you get a profile, a parabolic profile, because the protein would be held back on the sides of the capillary. Now, this is quite normal in any capillary, but it means also the particles, they diffuse out of the center, right? So, and at the end, they come back here. And if you monitor that from the side, your indicator would, the traveling of your indicator through the capillary results in this profile you see on the top of the fibril. And if you now change the size, diffusion is size dependent. If you change the size, basically the parabolic profile becomes wider and wider. Yeah. So, and this means the plotting of the parabolic profile gets also what you see on the right, wider and wider. So, based on this, as diffusion is depending on the hydrodynamic radius, or the apparent hydrodynamic radius, you can actually get a readout for the size of your molecule. Now, that's actually quite interesting when you want to observe the size of aggregates, because actually if you have just a small particle, you get quite a narrow parabolic profile. Now, if your small particle binds to something big, right? For example, instead of the monomer, it binds to an aggregate, you get a wider profile. Now, this is something you can plot. And when you plot these data, yeah, you can, this actually, what you see on the left is basically how the data look like, yeah, and you get actually, you can square of the width of the peak actually is in reverse correlated with the diffusion constant, and the diffusion constant again inverse correlated with the hydrodynamic radius. So, and based on that, we now can characterize actually fibrils. And one example I would like to show you here is the aggregation of tau over time. And what we do see here, right, we have on the right panel, we observe tau aggregation over 12 hours with a classical THT fluorescent assay, which I've shown you before. And what you see is the aggregation of the tau protein. So now we can take samples and measure in this FIDA instrument actually the hydrodynamic radius in nanos, should be nanometer here actually. What we do see here when we measure that in nanometer, we see that the size on the right simply increases over time. And this means we now have a tool to characterize the size of these fibrils and actually, for example, test the chaperone activity on our fibrils. And this is something we are now, I'm afraid it's too early to show you the data because we are not done with that yet, but I mean, we actually have now characterized and measured the size of the Alzheimer fibril, of the PIX fibril, and we end up there with the size of, say, around 50 nanometer, and that's actually quite reasonable range of fibrils. And with that, we now are able to test the effect of chaperones on these fibrils. So basically, based on the data I've shown you, it seems to be that fibrils are too late for chaperone action. And when we now think about what does it mean for chaperone action, and also what it would mean for drugs, it would mean you want to act early. You want to act on early intermediates. And so now that's basically the message, what we need to look for. And I think we now have the tools to do so. Good. Finally, I would like to thank people who did the work. I mean, the work on the chaperone mechanism had been done by my former lab member, Tania Moran-Luengo, and the work we have now presented on tau is in particular the work from Julia Achnes-Petroler and from François Decker, who now also works with the FIDA more on Huntington. And the project and work with the patient fibril was actually started by Mechet Koopman. And I do like to thank my collaborators, in particular now Matthias Meyer, with whom we collaborate on the HP70 system with a group of Friedrich Förster, which we collaborate on the EM on the fibrils. And from George Harris, we learned how to purify the protein from a patient brain. And I would like to thank you for attention.
Thank you so much. That was extremely interesting to hear about the interactions of the chaperones with disaggregating the proteins. Let me see if there's no Q&As yet coming, but I have a few questions for you while we are waiting. Can you comment on the crowdedness of the cellular environment? Do you think it will have an effect on, like, it will change the active concentration of HSP17?
Oh, in fact, sure. I mean, crowding is a real effect, no? And it does influence that. And I did show you data from firefly luciferase, no, where we did unfold and measures refolding. Yeah. Now, what you can do is you can measure the activity of luciferase also in the cell, in bacteria. No? Okay. So, which means you can heat shock the bacteria and the luciferase unfolds in bacteria. And then you can look as, okay, what is now the refolding in the presence or absence of HSP9? In fact, how we did it in the present absence of HSP90 inhibitors, and then you can reproduce the same effect, which we now have seen here in vitro. You can basically observe in vivo as well. So, I mean, obviously, chaperones, they are developed, evolved, and optimized for acting inside the cell, which means acting inside the crowded cell, right? So, can say, well, it's more then surprising that they also do work in diluted solution, which they do, but for sure they do work inside cells.
So my question was, that explains it, that the active concentrations, the active can be like different from the aqueous from the cellular environment.
Well, I mean, in fact, the concentration in the cells are very high, yeah? But you have also different environment. And I mean, the effect of the macromolecular crowding is that diffusion of all particles is much slower. No, that's
so we should.
And you also change. You on the one hand you may favor aggregation, but you also, due to the lower diffusion, you counter aggregation. So it's a complex situation due to the crowding, but also you have a lot of partners that may participate in aggregation events, may trigger aggregation events, which you do not have in diluted solutions. No, but clearly what we see is the effect and the activity of these chaperones. It's basically it's the same in diluted solution and inside cells.
Yeah, so that's really reasonable. And we have a question coming from Sunil. Nice talk. I'm curious if you comment on the mechanism of action of chaperones on different fibrils, means how specific the effect of chaperones on different kind of fibrils. Is it structure specific or sequence specific effect of the chaperones?
The very short answer is that I have no clue. And so, because I mean, in fact, what is quite interesting that the major proteins that do form protein fibrils, such as tau and all these tauopathies, such as alpha-synuclein aggregating in Huntington's disease, or in Parkinson's disease and Huntington aggregating Huntington's disease. I mean, all these proteins, they are known to interact with HB70 and HSP90 as monomers. Yeah. So actually, tau under normal circumstances, tau is degraded via an HSP90 dependent pathway. So basically, what HSP90 does, it interacts with ubiquitin ligase, and via basically, instead of, let's say, folding it, tau essentially disorder will never fold. No? Instead of folding, it basically gets a kiss of death from interacting with HSP90, because it basically HSP90 recruits ubiquitin ligase, ubiquitinates tau is degraded. So that happens to the monomer of tau. Good. So now we wonder, when you have these various aggregates, the monomers do interact with the chaperones. What would, when would the aggregate with fibrils? Now, we have looked at, in particular at, we have looked at 3 tauopathies with very different structure. From patient, it doesn't seem to have an effect. I mean, actually, I should say that the laboratory of Carmen Nussbaum-Kramer and Ben Bucher, they have seen an effect, but they did sonicate the fibrils. And then you have very different things than if you have actually fibrils just isolated from patient maturity, and you have basically a fibril which you still can see in EM. If you sonicate them, you can't anymore. So it really depends definitely on the size of the fibril, I would say, that you can disaggregate small, artificial fibrils with a small interface, but you, in our hands, so far, we don't think we can aggregate the patient fibrils.
Okay, yeah, thank you so much. You have another question coming from Becker too. Do you find which FIDA early intermediates of defined size that are stable for a while and would have to refold into filaments, or is a FIDA indicating continuous growth from monomer to filament?
I mean, we hope that we are able to characterize that. Yeah. So in principle, the technology is able to actually to give you the size of a particle between 0.5 nanometer and 1,000 nanometer. So this means that all stages of the aggregation process are in there. Now, your question is actually very good because you ask, can we stabilize the oligomers, right? That's the million-dollar question, billion-dollar question probably, multi-billion-dollar question. Could we stabilize relevant oligomers? And well, we can't answer that question yet. I mean, obviously, if we can stabilize them, we can observe them and measure them, and then we can see what happens to them. But I can't answer that question yet.
Yeah, thank you. And I kind of have a follow-up question about the FIDA. What is the resolution of this technique? Like how big of a differences you can see from particle to particle?
Well, I mean,
you said the range, but like, can you distinguish like 20 from 50 nanometer and so forth?
Well, I mean, I've shown you the data we had now for what we now measured for tau, yeah? So the answer is yes, you can. I mean, see, it's an experiment, no? So if you inject samples, so what you typically do is that you also see that you shoot in several samples and then you plot that. But I mean, in the data I have actually shown you, it was basically a growth from the size of a tau monomer, which is is around 1 nanometer, actually to fibrils up to 60 nanometer. No, I mean it's and you see that growth over time. So yeah, in principle, it's suitable for that range. And the great thing is when you work with patient derived material, you need very little. No? For a shot, you need 40 nanoliter. And that compares quite favorable to many other techniques.
Yeah, thank you. That's this technique is really interesting. How does it look like? It's a microcapillary, I assume. With like a attached spectrophotometer, right?
You could say, well, it doesn't look like a black box simply because the box is blue. But when you open it, there's a capillary inside, yeah, and then you can say, well, it's actually quite simple, no? That you you just measure the diffusion of a particle through the capillary, and the art. What you need to do is you need to fluorescently label your sample, or you have a fluorescent dye that actually binds to that what you want to observe.
Yeah, that's a big caveat. Fluorescent labeling.
Oh, it's like caveats, but
no caveat, but yeah, you need to deal.
No, I mean it's really also a part of the challenge here, because we now this took us quite some time that we managed to get a readout for the fibrils, right? So that you only see the form fibrils, but you wouldn't get a background from a free dye or things like that. Yeah.
Yeah, that's really cool. So my other question is, it's kind of more your comment. There are some chaperones which actually, in fact, their action is connected with the increase of the aggregation, and they produce more oligomers. What are your thoughts about that?
Well, I mean,
that has been shown for the yeast chaperones, like in the case of yeast. For humans, I don't know which one is the equivalent.
Well, I mean, we have seen ourselves with HSP90 that this HSP90, on the one hand, you can prevent aggregation of tau. But we did measure that with EPR in collaboration with Malte Drescher's lab. But but HSP90 also somehow facilitates the formation of an tau aggregation nucleus, and that potentially by providing orientation of tau that facilitates fibril formation. So, I mean, in principle, it can work both ways. Huh? So, on the other hand, I think it's it would be, in a functioning cell, you shouldn't expect that actually the chaperone machinery itself should produce constantly fibrils. Otherwise, no one in the audience would live long enough to participate in listening to this talk.
Yeah. Yeah. Thank you so much. Yeah, that was really, really interesting.